Okay. How many are we normally? Is this about the group? Yeah.
Yeah.
Okay. All right. Well, yes, we'll go ahead and get started. So we're really delighted to have Dr. Bridget Barker here as our guest speaker for our EvoFunPath seminar. Bridget, I think hopefully you were briefed a little bit on the program, on the EvoFunPath program, and kind of who we all are on this end. So I'll introduce Dr. Barker, who received her BA in biology and MSc in ecological genetics from the University of Montana. Dr. Barker started her PhD program as an Eigert Fellow at the University of Arizona in 2004. This is where she became interested in working with human fungal pathogens, and specifically on Coccidioides, Coccidioides immitis and posadasii.
Yes.
Yes, got it. Which you're going to hear all about today. So these are the causative agents of coccidioidomycosis, also known as valley fever. And so in 2009, Dr. Barker completed her PhD with co-mentors Dr. Scott Crokin and Dr. Stephen Rounsley, and then started her postdoc work at Montana State University, where she shifted gears to working on Aspergillus fumigatus in the lab of Dr. Robert Kramer. So as of 2016, she started her position where she is now at Northern Arizona University, NAU, in Pathogen and Microbiome Institute. And as of just 2020, she's now an associate professor in the Department of Biological Sciences there. Yay.
I'm still celebrating.
Yes, I think you get like 5 years of celebration easily.
Yes.
And so Dr. Barker has really extensive experience in genomics, bioinformatics, population and molecular genetics, and evolutionary biology. So we think she's going to be a really great fit for this program and for you to all hear from. And we're very excited to have her here today. So please join me in welcoming Dr. Barker.
Thank you so much for that very kind introduction. Yeah, so I've had a wide and varied career, and I will talk kind of broadly about the organisms that I work on, and I will put it in both an evolutionary and an ecological context, and share some new unpublished data with the group. And hopefully you are as excited about it as I am. So share screen and we will get started. Great, everything look okay? Perfect. So today we'll be talking about my favorite organisms, Coccidioides immitis and Coccidioides posadasii. So these cause a specific disease. Coccidioidomycosis is the medical term, but we call it valley fever because it's much easier to say. I will frequently just refer to the organism as cocci. That's sort of shorthand because it just makes everyone's life easier. So in my lab group, we focus, I said, primarily on the ecology and genomics of Coccidioides immitis and posadasii, but we do dabble in the other medically important nigonales. We've done some work with histoplasma, paracoccidioides, and are sort of emerging into these other relevant organisms, blastomyces, amansia, and emergomyces. There we go. So again, to orient you to the group of organisms that I work on, this is a very old tree. I've been working on updating this slide, but I haven't quite gotten there yet. And really all I want you to focus in on is the fact that Coccidioides is a member of the Ascomycota in the order Anigonales. So this is a very interesting group of organisms for a number of reasons. One is the fact that I get to show very interesting pictures of what these organisms do in the environment. So these organisms really specialize in breaking down keratin and animal-associated proteins. So here we can see specifically these mushrooms, basically mushroom-like sexual structures that are forming on the horns of a sheep, bison, the hooves of horses, etc. So these are commonly found associated with these types of decaying material in the environment. but also these organisms cause a lot of disease. So the order Nigonales is most famous for these pathogenic organisms. So again, blastomyces. There are probably other hosts besides humans, but it hasn't been very well described. In the pathogenic phase is yeast-like structure. Histoplasma, we know, has a pretty predominant association with bats, also growing as a yeast-like structure. There are other organisms within this group named Amansia, although again, this is in the process of being renamed. So this is probably Amansia eleopsis, and Amansia here is probably part of the group that's actually going to probably be renamed Emergomyces. But both of these structures here, these large adiospores, are somewhat similar to what we see in Coccidioides, and I'll show you some specific pictures of that. These organisms, as I said, break down keratin, other fibrous structural proteins associated with animals. There's a lot of genomic support for their ability to digest these animal-derived materials. They are capable of colonizing both living and dead host tissues and cause disease in a wide range of animals. Also important, again, for that evolutionary context, this is sister to the Uroshiales. So the Nigonales ranch here is sister to the Uroshiales, which contains many other relevant human pathogens. So Aspergillus fumigatus being the one that's represented here. And broadly, this whole group is contained within the class Uroshiomycetes. So as you notice from that picture, from that phylogenetic tree, there are two species, Coccidioides immitis and Coccidioides posadasii. So we knew that for a long time based on genetic profiles. However, there was no real phenotypic difference that had been observed between the two species. So a talented PhD student in my group, Dr. Mead, worked on trying to identify some observations that I had seen previously and really follow up and make this a very nice, thorough study. So we found that on nutrient agar plates, posadasii was able to grow, albeit slower than at 28, was able to grow at 37, whereas immitis grows very, very well, in fact, better at 28 degrees than posadasii, but at 37 degrees, it is very inhibited. And so we generally see very limited radial growth on plates. So this was the first, and to my knowledge, only phenotypic difference that's been investigated for Coccidioides. The life cycle is really separated into the saprobic life cycle. This is what we find most frequently in the environment, and the parasitic life cycle, which is what occurs in hosts. So again, this causes coccidioidomycosis or valley fever. The disease is very complicated, and we'll get into some of the statistics there, but basically the organism can disseminate to pretty much any tissue in the human body. So it's been found in joints, there's oftentimes bone lesions that are associated with that. There can be skin ulcerations, and in the most extreme case, becomes meningitis. The organism can probably form a latent phase. So there are many cases where there's emergence of disease. So a patient has a diagnosis and 20 years later has an emergence of more severe disease later in life. We believe that's associated with various immunocompromising conditions, but it hasn't been very well investigated, and there's not a animal model for that. So mice are very susceptible to infection. So we don't have any chronic models for this disease. Immunity, there's cellular-based immunity in humans and mice. Unfortunately, the early innate immune response has not been well characterized. So we really don't understand what happens in those first few days of infection. So when those spores are inhaled from the environment, they take about 4 to 5 days before we start to see the maturation of the actual spherular-like structure. So I'd shown you the adiospores, so that's more similar to this. structure here, but for Coccidioides, and to our knowledge, Coccidioides is the only group of organisms that does this, has an endospore phase. So they actually form endospores internally to the structure. Those endospores mature, the spherules ruptures. Each of those endospores can then become a new spherule. So fairly quickly, the host can become overwhelmed, and these basic first time periods of the infection are not well described and not very well understood. So some statistics about infection and case numbers. So we really don't know the actual number of new infections annually in the United States. So these are the reported cases of the disease. So in blue, we have cases in Arizona, and in orange are cases in California. So this disease occurs throughout the southwestern United States, but about half of the states in the United States do not actually report the disease to the CDC. Even though this is a nationally reportable disease, it's still up to the states to report that to the CDC. So the reason that we kind of extrapolate and try to understand the actual numbers of new infections is because we really want to have a better understanding of what's the actual disease burden throughout the United States. So if we think that only about 10% of the cases, the infections actually get reported, that would mean that, say, in 2019, there were about 18,000 cases reported, again, only from California and Arizona. So if we take those numbers out, we think that they're probably anywhere from 180 to 250,000 actual infections every year in the United States. But again, we don't really know, and that's just our best guess at this point. So where do we find the organism worldwide? So it really has only been identified in North and South America. So in the United States, that's going to be, again, primarily California and Arizona. In California, we find mostly Coccidioides immitis. In Arizona, we find Coccidioides posadasii. Texas, New Mexico, Utah, Nevada, and Eastern Washington are all states that have reported fairly significant case numbers. Although, again, Texas does not report those case numbers to the CDC. Based on patient isolates, we have identified these states as being predominantly Coccidioides posadasii. And the small region of Eastern Washington has been identified as predominantly Coccidioides immitis. Again, we sort of extrapolate out based on sporadic case reporting that pretty much any arid region in the Western United States is potential habitat for this organism. In Mexico, it's also fairly complicated. We don't have reporting from Mexico at all. These are really based on fairly old data from skin testing and when the disease was reportable in Mexico. In Sonora, we find both immitis and posadasii. Baja, California is predominantly Coccidioides immitis. Michoacan, which would be on the West Coast, is Coccidioides immitis and posadasii, and everywhere else that we've looked, it seems to be Coccidioides posadasii. And then again, sporadically throughout Central and South America, caseloads in Guatemala and Honduras are not very well characterized, but the few isolates we've had indicate posadasii. Colombia is interesting, a mix of immitis and posadasii. And then for the remainder of the small number of isolates that we've gotten, those have all been identified as posadasii. So this is an environmentally acquired infection, which is why I spend a lot of my time thinking about the organism in the environment. So we have infections that result from direct inhalation of the arthrochonidia in soil, dust, air. So really understanding ecology is important. So trying to understand what that environmental reservoir is will help us prevent infections in the first place. There's no real evidence for transmission from an infected host other than some very rare cases of organ transplant. And I'll show a little bit of a story on that. The data on the environmental niche is pretty limited, which limits our further ability to do climate modeling and our ability to predict when and where we would assume that people are getting infected. The pathogen infects healthy mammals, and there's some evidence that it could be infecting reptiles, although that data is also extremely scarce. So from my brief introduction of where we find it, we quickly realized that there are fairly strong biogeographic patterns among the clinical isolates. So again, we really only have investigated isolates from patients. Patients move, and so it's not 100% identification of where the patient actually was infected. So again, we believe that immitis is more predominant in California, down into Baja, Mexico, but there's some overlap, probably where those ecological ranges mix. This is work that was done by a postdoc in my lab looking at isolates based on PCR. So these are 9 microsatellite loci that we characterized and found this confirmation of a biogeographic pattern. So of course, you know, microsatellites aren't definitive. There's a lot of issues with analyzing the data that way. So we proceeded to actually do whole genome sequencing on, we're up to actually over 300 isolates currently. We're waiting on a last bit of data from isolates from Brazil. And then we hope to do a final full phylogenetic analysis across the entire biogeographic range of the organism. But for now, the patterns seem to be fairly consistent. And so we don't expect any major changes other than probably resolving this clade here. So again, I'm in Arizona, so I have access to a lot of isolates from Arizona. Interestingly, when we subject these isolates to phylogenomic analyses, we actually have a distinct set of genotypes from Tucson and Phoenix. So for those of you not familiar with the geography of Arizona, Tucson and Phoenix are separated by 100 miles. So they're only 100 miles apart. I can't convert that to kilometers, but hopefully you know that they're not very far apart, but they are ecologically very distinct. So Phoenix would be characterized by a lower Sonoran life zone versus Tucson being the upper Sonoran desert biome. So higher elevation, cooler temperatures. Phoenix is going to be lower elevation, hotter temperatures that could affect both the organism itself, and then also if there are specific hosts that these organisms are adapting to, it could also be reflected in this pattern. So that is further analysis, but an ongoing, but it's an interesting pattern nonetheless. So what can we do? You know, what are some other reasons why we want to understand the phylogenetic landscape of the organism? And that's partly to conduct investigations like this. So this is a case where a very young child was infected and diagnosed in New York, but had travel history to both California and Texas. And the region in Texas that they traveled to is not a region of Texas that is commonly thought to have the organism in high prevalence. So this is more on the eastern part, very close to Dallas, actually. And so not a region where we think of having a lot of Coccidioides in the environment. Indeed, when we did the phylogenetic analysis, this was traced to the Texas. So this here highlighted in yellow is Beeville, which is the closest town where this child had stayed with their parents and grandparents. And this groups very clearly with Texas, Mexico, South America clade, not with an immitis, which would have indicated the child was infected in California. We can also use this information to understand a case like this, where there was a cluster. So this was patient X, Y, and Z in Los Angeles, California. What united these patients is they were all recipients of transplanted organs from the same donor. And so because Los Angeles would be in the endemic region, they wanted to understand whether or not these patients had gotten valley fever because they were immunosuppressed and had all gotten different exposures, or if this was actually a case of the infected organs being transplanted into the patients. So in this case, the evidence is pretty clear that these all had the same source. They are clonal, and these patients were actually infected by the infected transplanted organs. So this changed how this institution did going forward, screened the donors for infected tissues. So that's information on the understanding the genotypes, understanding the biogeographic structure, some of the things that we can do with that information. But what I've been really interested in exploring is trying to understand the genetic variation that links to phenotypic variation. So what we might be interested in is, are there differences in pathogenicity? So diagnosing valley fever really doesn't require that you understand whether it's Coccidioides immitis or Coccidioides posadasii that's causing the infection. But we don't really know that without doing that diagnostic, we don't understand whether or not there are potential differences between the two species in pathogenicity. Variation in severity based on the type of isolate that you're infected with is not very well understood or characterized. Additionally, we don't have good information on whether or not there might be differences in treatment, right? So vaccine targets, as we develop vaccines for these diseases, making sure that we have cross protection and good protection for both species. Also the potential for variation in antifungal drug resistance. So these are areas where we're actively exploring using some of this phylogenetic information, as well as epidemiology, right? So possible different associations with different hosts in the environment, possible different outbreak patterns. So different times of the year where immitis might be more prevalent in the environment versus different outbreak patterns that might be associated with posadasii. So this gets to this idea of reverse ecology. So understanding genetic variation might help us understand the ecological niche. So what do I mean by that? So this is just an example of a type of analysis that we've done. Obviously, this is ongoing work, but this was published with the first genome paper. When we looked at the coding regions for various plant-associated or animal-associated genes, we found an expansion of keratin degradation. So acetylysins, peptidases, metalloproteases, et cetera, are those types of enzymes that are associated with breaking down animal material. And these are widely expanded within Coccidioides. On the left here, we see what we would consider to be plant-associated gene families, so tannases, pectin lyases, et cetera. And these would be genes that are associated with the breakdown of cellulose, other plant-associated compounds. And for posadasii, we see a loss. There are no genes that are associated with those particular functions, and a reduction when we compare it to its close relatives. So these are the Aspergilli, which would be that sister Uroshiales order, and these are the Onigonales, Histoplasma, and Ensidocarpus, that would be in the Onigonales and associated more with the breakdown of animal material. So what are we doing to go forward with this? So how are we linking genomics and these genomic profiles with ecology? So we're re-annotating a lot of these genomes. We are trying to do a better job of assembling genomes and produce reference genomes. So we just recently published on a type strain for Coccidioides posadasii and basically did a telomere to telomere assembly of that genome and did a reannotation of that genome. And so going forward, we're going to continue to do this for multiple genomes and really try to determine gene gain and gene loss, both within the onigonales and also in between the two species of Coccidioides. We want to identify potential gene family expansions. So we also need to do better sequencing of close relatives. So that's ongoing work. We are also looking at genes that might be under positive selection. So again, once these annotations are complete, we will do a thorough analysis of genes that might be under positive selection, which could be novel antigens. So things that might help us with diagnostics, things that might help us with vaccine development, et cetera. This is going to involve a lot of expression of transcripts. We're also interested in looking at these transcripts in vivo. So we've done a few small studies where we've done RNA-seq on lungs from infected mice, as well as nanostring, and some interesting patterns have emerged. But usually this is just one or two isolates with a few time points. So these are things that we really need to expand. So I'm going to switch a little bit to focusing on the ecology and then discuss at the very end some new findings. So one of the things that was very clear to me when I first started looking for the organism in the environment is we really needed a higher throughput way to find cocci in positive sites. So the previous molecular methods were not great. And culture-based approaches is extremely labor intensive. And actually, we're finding that for posadasii, we can't easily do direct culturing for reasons that I will leave as a tantalizing question for you to ask later on. So we developed this assay. We called it Cocci-ENV for environment. It's a TACMan probe-based real-time PCR. If you're interested, this is the publication. It was in medical mycology. And we now have this tool that we are applying to detect the organism directly in the environment. So what do we understand based on the clinical information? What are the regions that we find cocci? What are some characteristic features of those areas? So the Sonoran Desert is a very special environment, and it's also occupied by many other endemic species. So endemic plants, endemic flora and fauna are very unique to this region. So many species are only found in North and South America. So armadillos would be one example, various reptile species, and a whole group of rodents called the heteromyids, which are very common in the desert environment. So heteromyids would be organisms such as pocket mice and kangaroo rats. And then obviously other microbes. So this hasn't been investigated very deeply, but there are probably many other microbes that are only found in this region as well. And then thinking about abiotic differences. So these are desert soils that are characterized by very specific types of biological soil crusts that grow and basically hold that desert soil intact. These are highly damaged and impacted by grazing and agriculture. So I have a master's student that's specifically focusing on biocrust and interaction with cocci. It's very interesting already. The pH of these soils is commonly more alkaline. There are patterns of seasonal moisture with very long periods of drought and extremely high temperatures. So there are many biotic and abiotic factors that probably influence the growth of cocci in the environment. So one of the questions that we had is, is the host itself the preferred environment, or is it the actual place that they live in the environment? That's the preferred environment for cocci. We asked this question because rodent burrows often represent point sources of infection in outbreaks. And when we go out and sample the desert, a majority of our positive samples are associated with burrows. So this is another recent publication by a PhD student in my group. that actually specifically investigated the role of animal burrows in Arizona soils. So the evidence that we have that animals play a significant role in the environment, whether that's the burrow or the animal itself, is this strong correlation between the presence of coccidioides in the environment and the presence of a burrow. So we had 131 total samples from 3 different regions, Lake Mead, Tucson, and Flagstaff. 36 of those were positive for cocci. If we break that down between burrow and non-burrow, and so these were soils that were collected within a meter of that burrow site, 25% of our samples were positive when they were associated with burrows versus less than 1% near burrows. So this is highly significant and indicates that burrows do indeed play some role, whether it's the host itself or the burrow environment in the presence of cocci. This has led to many interesting conversations, and I published recently with John Taylor on this idea of an endozoan-based life cycle. So we know that there are plant-associated fungi that live inside host in the plant host without causing disease. And so why couldn't this be the case for fungal pathogens or fungi that are able to live inside plant hosts without causing obvious disease? So this small mammal reservoir hypothesis is tantalizing, although we don't have specific proof at this time. It's an ongoing concept and an ongoing area of study for our group, as well as Dr. Taylor. The basic concept is that the sphereal itself would be an evolutionary dead end, right? So we acquire this disease directly from the environment by inhaling those conidia. So it's not transmissible. It doesn't get passed from host to host. So somehow this organism has to get back into the soil to undergo that saprobic life cycle again. So this could be either spherules develop and the host has a short life cycle, it has severe disease and dies shortly after infection, or potentially that latent aspect of the disease, this spherule can become a granuloma, and the host dies at some point, and that organism can still make it back into the soil. So in either case, we have a living mammal that gets infected, a dead mammal that re-inoculates the soil and initiates that arthrochonidia phase and is reinfected, or animals are reinfected, and that life cycle can continue. So to answer these types of questions, we're doing a lot of long-term monitoring of positive sites. So these sites do remain positive for decades. A lot of the isolates that we obtain from a single site are clonal, but patient isolates are mostly different genotypes. So there's a little bit of an issue with trying to understand where people are getting infected. Are there multiple sites that we haven't yet identified? Or is there something else going on that we haven't quite identified yet? And I will get to that in the next few slides, what we think might be going on. So we're really working on developing these methods to assess whole genomes without culture. So now we have many of these sites. We can develop now DNA probes based on the whole genomes that we have sequenced, and potentially pull out the DNA directly from those DNA soil samples, because we really want to understand how those genotypes might change over time, and how. So the how is the interesting part. So what other clues do we get about the life cycle and ecology when we mine those genomes? One of the things that has popped up frequently, and as we sequence more and more genomes, probably about 5 to 10% of the genomes are what we think are hybrids. So we have some evidence that immitis and posadasii can hybridize and potentially form viable offspring. So what are the implications of that? So that would suggest that Coccidioides is capable of mating. So it has both mating type loci, I'll define that in a second, and meiotic genes, but a sexual cycle has never been observed. So when I showed you that life cycle, it was all asexual development. So in the environment, growing as a mycelium, producing asexual conidia, in the host, growing as a spheruole, producing asexual endospores. The two species have generally synthetic chromosomes with a few minor differences. The species are separated by about 5 million years of evolutionary distance. So the existence of hybrids would suggest that sexual recombination is occurring. So what are some advantages of sexual recombination? So obviously, it's going to provide a diversification in natural populations of fungi. You're going to generate novel genetic variation. You're potentially going to produce individuals with altered virulence, increased fitness, potentially in new ecological niches. And those new ecological niches could be either new soil types or new hosts. So novel hosts, that idea of potentially switching between humans and dogs and desert rodents, how can this organism potentially infect all of those different hosts? Well, sexual recombination might be one way that could happen. And of course, there's the classic purging deleterious mutations from the genome, which is obviously important as well. So we have this question, is the variable genetic background responsible for potentially having more severe disease in healthy people? So occasionally we have a patient in the clinic who has no other obvious immunocompromising situations, but is extremely ill. And in fact, in a few cases, the person dies very quickly from the disease. So is this potential, is this host potentially infected with a novel, highly virulent isolate? The new genetic background could potentially allow dispersal and colonization of new environments and new hosts. So understanding gene flow and admixture, we really want to understand if there's either a sexual or parasexual reproduction strategy and recombination that might be explaining this gene flow and admixture that we have observed. So our approach to understanding this is very old school, and I will show you many, many beautiful images over the next about 10 minutes, I think I have left. Yes, okay. Oops. Okay, so mating type loci. These regulate the sexual cycle in fungi, and we think of this as the sexual identity. It's not male-female, but it's an identity, and these are based on 2 transcription factors. So MAT1-1 is an alpha box, and MAT1-2 is the HMG high mobility group transcription factor. And I won't go into any more detail than that for now in the interest of time. But basically, we've looked at these mating type loci. So another point that you need to be aware of is cocci is haploid. So it's either MAT1-1 or MAT1-2, because this organism is also homophilic. So it has only one or the other. It can't, as far as we know, self-mate and has to find a partner in the environment. When we've looked at the distribution of these 2 mating type loci, we generally see it in a 50-50 ratio for most of our populations. We do occasionally find it in rare cases in Venezuela and in Washington, so both posadasii and immitis, where we only see MAT1-2. And so that is an interesting observation, but these are fairly low numbers, so N of 7, N of 6. In this region, and the significance of this is still, we're still kind of working through that, but for the most part, in most of these populations, we see these mating type loci in a 50-50 ratio. So a very talented postdoc in my group, Marcus Teixeira, investigated this and did some of the first mating crosses. So here we've placed the organisms, so 2 organisms on the same plate. And we started to observe these very interesting coiled hyphae, which are indicative of what we call young ascocarps. We see similar structures, again, in close relatives, Paracoccidioides and Histoplasma capsulatum. We see similar coiled structures. So again, possibly some signals that the organisms are giving each other to initiate mating. So again, what we see on a plate are these small little tufts of organisms. We pull these off and investigate them, and we see these coiled hyphae, again, indicative of what we call young ascocarps. So the initial developing structures associated with sexual recombination, similar to what we would see in close relatives, Histoplasma and Ansinocarpus here. We see what are called cleistithecium-like structures. So again, these coiled balls of mycelia, we take those and investigate those microscopically. We see these structures with the coiled hyphae, again, very similar to cleistithecium-like structures in a close relative. So we wanted to take a closer look at this. So that was just light microscopy. What happens, what will we observe if we look at these with electron microscopy? So again, very distinct, highly structured cleistithecium-like with lovely coiled hyphae. Again, very, very similar to the sexual phase of Histoplasma capsulatum. And in a few cases, we observed structures that we would consider to be what are called asci. So the asci are going to hold the ascospores, which are the structures that are the products of meiosis. And similar structures were observed again in Ansinocarpus. However, we never observed the actual ascospore-like structures. So we have everything indicating that sex is happening, but no final structures associated with that. So it was time for Marcus to move on and go back to Brazil and be a faculty member. So I have a student now, Claire Laux, who has taken this on. And so she has continued this work. And again, here is a lovely picture where we have one of the mating types in the center, surrounded by the other mating type, forming these characteristic ridges in between the two colonies on various types of media. So again, she did a lot of work trying many different types of media so that we could have these structures observed more frequently and potentially observing what we think might be ascospores. So the previous experiments, those were months-long experiments. Claire's gotten this down to weeks-long experiments, and we start to see these structures. So this structure here is again consistent with the ascus. So these tufts, again, looking at these coiled hyphae, zooming in, here is our first indication of an ascus. So this vase-like vessel with spores internally. So some evidence of cell fusion accompanied by this very characteristic structure. And you can see several ascospores here, which are very distinct and unique from the Arthrochonidia, which are barrel-shaped, and these structures are clearly circular, disc-shaped structures. So a greater number of ascospores and asci are now being observed. We have reliable methods to produce these in a short period of time. Here you can actually even see an ascospore being ejected from the ascus, which I don't know. To me, it's very, very exciting and very satisfying. Now we're working on methods to reliably harvest those ascospores, grow them, and characterize the recombinant offspring. So again, just to kind of briefly lay out what we've observed. So these are all of the structures that we've observed in Coccidioides. They're very similar to structures that have been observed for blastomyces and histoplasma, both of which have a defined sexual life cycle. So now we want to understand the significance of that. So as we are able to harvest those ascospores, identify those novel genotypes that come out of them, we want to understand if those potentially could be the environmental reservoir. So these ascospores have very thick walls. They potentially can live for much longer periods of time dormant in the environment. They could also have the ability to infect novel hosts and expand host range, and infection with an ascospore could cause differential disease. So in the environment, if we do have situations where there are both arthrochonidia, the asexual structures, as well as the sexual structures, and a host gets exposed to both at the same time, what are the consequences of that versus only being infected with conidia, only being infected with an ascospore? And those are all ongoing areas of investigation. Some big questions that we're trying to answer is to really understand and characterize these soil sites. So again, understanding what's going on at the soil sites might help us answer that question about whether or not there are only clonal asexual conidia at most of these soil sites, or whether we have some evidence of some recombining genotypes or potentially a mixture of both of those present at the same site. We want to look more intensively at patients and clusters of patients that have similar genotypes. So we're working now currently with Arizona Public Health to identify potential clusters of infections and then obtain those isolates from patients where they're available, and then genotype those and understand whether or not the patients that are geographically clustered have similar genotypes that they are infected with. So that would help us to ID hotspots of infection. So obviously a lot of people to thank. I have a great group of technicians and undergraduates and graduate students that help get all of this work done. I've highlighted the two that have done most of the work. So Marcus also was responsible for a lot of the phylogenetics. We actually collaborate quite closely now that he's a faculty member. We collaborate quite closely on the investigation of specifically isolates from Brazil right now, as well as other isolates from South America. And then Claire, who did a lot of that new work that I've presented on identifying the sexual cycle. And with that, I'd be happy to take any questions, and thank you for your attention.
Let's do